Hi everyone, I am Chloé Bars and I am a PhD student within the Comparative Perinatal Development Laboratory, or COPED Lab, at the University of Antwerp in Belgium. I am excited to share with you our research interest towards the characterization of the zebrafish embryo model, a promising alternative for developmental toxicity assessment. More specifically, my PhD project was dedicated to unravel the potential xenobiotic biotransformation capacity of the zebrafish embryo during its whole period of organogenesis, and therefore its capability to mimic mammalian condition during drug screening assessment. I am excited to further exchange with you and discuss with you towards this subject. Zebrafish embryos have many advantages. Among others, it is not considered as an animal model until 5 days post-fertilization under European regulation. Therefore, it is an in vitro model while being a whole organism. It also has a transparent eggshell, which allows us to fully observe its development just under a microscope. To finish, a female can lay around 100 to 200 embryos per spawning, so we have a lot of sample to conduct our experiment without harming the mothers and the embryo. It is the process by which an initial mass of embryonic cells slowly differentiate into the different complete organs of the embryo, such as the heart, the kidney, the livers, for example, but also at very specific position in the embryo. While amazing scientific discoveries brings the answers to the question of nowadays, with the development of new techniques, compounds, and molecules, I have been drawn on the other side of those discoveries and to ensure the safety of humans and the wildlife. However, those new discoveries could also help in the development of alternatives to animal models and by their integration in the testing process. The latter has a bright future and is very stimulating. We are focusing on compounds called proteratogen, such as the antiepileptic drug carbamazepine. In humans, those compounds are not embryotoxic by themselves, but by their toxic metabolites formed by enzymes present in the mother. Therefore, to test the metabolization capacity of zebrafish embryos early on, those compounds are perfect because they need to be metabolized to induce embryotoxicity. Therefore, by just observing malformation, we know if the compound should be metabolized. We discovered that after being exposed to carbamazepine, the zebrafish embryos can metabolize it and produce the same metabolite as in human. However, the embryotoxic metabolite in human is not responsible to the malformation observed in the zebrafish. Therefore, the latter might be due to other metabolic pathways specific to the zebrafish. Therefore, the full characterization of this model needs to be continued.